Greetings. This is a presentation that I made to the American Association of Physicians of Indian Origin on the 6th of January 2022. This is COVID-19 Perspectives on Omicron, Vaccine and Epidemic Control. In this presentation, we will discuss why Omicron, why now. In the how, we will touch on aspects of virology, complexity in epidemiology, and in the what, we will touch on epidemic control, particularly what may work. So as I said, this is a recording of the presentation I made to the American Association of Physicians of Indian Origin, the Global Healthcare Summit 2022, on the 6th of January 2022 at Avasa Hotel in Hyderabad. A brief prayer, Loka Samastha Sukhino Bhavantu, in all of creation may joy happen and let there be tranquility. So projections for epidemic dynamics in India, what we really need to look at is a socio-economic perspective based on integrating basic science of virology, medical sciences, epidemiologic and historic trends. This is essentially a recapitulation of what I had stated before in my presentation to the American Association of Physicians of Indian Origin and the Chicago Medical Society last summer. In all likelihood, we will see that the epidemic numbers decline in the following few weeks, followed by an inevitable rise in cases within a few months of the decline. The scientific reason for this is the logistic map equation. The only significant change in epidemic dynamics is the possibility that we have the ability to vaccinate over 75% of the population, including children, within the next few months. This is a highly questionable probability because it is important to understand that children below the age of 14 constitute nearly 28% of the population in India. We have also seen that children easily acquire and transmit respiratory tract infections with mild illness in children, but serious illness when transmitted to adults. Both mycoplasma and chickenpox are classic examples. This has serious implications for schooling also. In India, relying on distance learning through the internet for many children is a fantasy. We do need to prepare to take care of the affected people in the community rather than in hospitals for three reasons. One, in India, 65% of the population is rural with negligible access to adequate medical care. Two, we do not have enough capacity in public hospitals to handle the burden, and private hospitals are not an option for the average citizen, even in urban areas. Three, concentrating large numbers of infected people in small areas, particularly poorly ventilated areas, which is what most Indian hospitals are, increases the risk of transmission to other people, particularly healthcare providers, and also increases the probability of emergence of yet newer variants of the virus that may be more contagious and capable of causing more serious illness. So we also need to have a plan to address not only the acute illness caused by COVID-19 infection, but also the economic impact of illness on the patient and family, socioeconomic impact of the lockdown, if any is applied, or any other social restrictions on economic activity. Long-term complications of COVID, particularly lung damage, the emergence of infections like tuberculosis and fungal infections, because COVID is a profoundly immunosuppressive infection. Long-term complications of inappropriate treatment regimens for COVID, in particular, overzealous use of high-dose steroids, overzealous use of immunomodulators like tocilizumab, and agents that really have no therapeutic value, such as remdesivir or ivermectin or convalescent plasma. We also need to consider the long-term disability caused by COVID and the consequent loss of ability to earn a livelihood. And this is in particular a serious problem in people of lower socioeconomic strata. Now, the Omicron variant probably appeared in late October, at least was first detected by the South Africans in late October, early November. Whether or not it had been in circulation for some time prior to the fact that it was detected in South Africa is an open question, because in the very nature of the emergence of variants, it is quite possible that variants emerge, are in circulation for quite some time before they are detected. Now, South Africa in particular has a rather good surveillance system, particularly in molecular surveillance, and therefore they were the first to pick up the emergence of this particular variant. Now let's see what has been happening in terms of Omicron as opposed to the Delta variant. We see that in South Africa, within a very short period of time, it has completely displaced the Delta variant. And even in the United States, by the end of December, more than 80% of the infections are Omicron rather than Delta. And India, in a short period of time, the numbers of Omicron approach more than 40% as compared to Delta. And as you can see in this slide, the number of infections are particularly high in major metropolitan areas, probably a consequence of better access to testing as well as greater opportunities for importing cases due to travel. Now, all epidemics, particularly very contagious airborne infections like coronaviruses, spread through contact, and this is the importance of understanding networks of spread. Very often we hear that somebody came down with an Omicron variant infection even in the absence of travel or even in the absence of known exposure to an index case. And the way this happens is somebody is exposed to an index case, that person exposes somebody else, and that person exposes somebody else. So this way you can get an infection from people remotely connected, even though there is no history of proximate travel or contact with a proximate traveler. So this is the notion of the 6 degrees of separation, and wherein we say that essentially we eventually are connected to each other. Now, the importance of understanding networks of spread is that in addition to an index case infecting one person, it is quite possible that there are super spreading events where one infected individual can infect multitudes of other people and vastly increase the number of people who are infected. This is a study done by Talbert and colleagues tracing SARS in Singapore, which showed that the virus was generally transmitted to few people, but there were super spreaders, some individuals who spread the infection to many, many people. Now, super spreading events have been noted in a lot of infections: Ebola, measles, pneumonic plague, SARS, and smallpox. Now, in some part, super spreading events combined with a very high R0 lead to even higher numbers of infected people. Measles in particular has a high R0 of 16, smallpox is around 5.5, and COVID-19 is probably somewhere in the neighborhood of 3, 4, in my opinion. This is a study that we did at Share India in mid-2020, where we used a rather simple model of susceptible, infected, and recovered, the SIR model, to do epidemic projections. And using this very simple model, making an assumption that the R0 would be 3 and the beta would be 0.7, we estimated that by late August, about 36% of the population of Telangana would have COVID-19 infection during this time. The basic reproductive number can be approximated as the product of the average number of susceptible individuals contacted by an infected individual during the infectious period, that is the number of contacts, and the average probability that a susceptible individual will become infected by a single infected individual during the infectious period, the shedding potential. So the R0 is a very important attribute of looking at epidemics and how epidemics will spread. Now let's see what actually happened as compared to our projections. In January of 2020, when the epidemic really started in India and also in the United States, we saw a fairly rapid increase, and it pretty much approximated our projections and perhaps even overshot some of our projections, which is why one asks the question, is the R0 really 3 or is it greater than 3? And if we look at the confirmed deaths, again, we see a similar pattern to the infections. Now, a word of caution, both infections and deaths in areas like India and South Africa may be subject to reporting biases because the surveillance systems are not quite as robust as United States. Although there may be other reasons for the difference in epidemic dynamics in South Africa, India, as compared to the United States that we will discuss a little bit later on. Now, if we look at the weekly epidemic dynamics, what we will notice is that it is a cyclical pattern. Not only that, please notice that India seems to follow the peaks of the United States by just a few weeks. Now, why epidemics become cyclical is something that we will discuss shortly, but I would also like you to notice the difference in the proportion of people infected, and why that happens is also something that we will discuss subsequently. The reason the epidemic becomes cyclical has to do with the logistic map equation. As R0 approaches 3.44949, the epidemic actually becomes cyclical with a doubling frequency or a frequency of 2. As the R0 keeps on increasing, the periodicity may go from 2 to 4 to 8 to 16 to 32 and so on and so forth. And as we can see from the previous graph, since the epidemic has been cyclical with a periodicity of 2, the R0 probably is somewhere in the neighborhood of 3.4494. The other thing that we noticed is the difference between the number of cases as seen in the United States as compared to India. And what this study shows is that differences between countries, as well as differences within a country between different counties, can be quite different, and they do not necessarily follow a simple increase or a decrease. In fact, the differences appear to show what we call a power law distribution. Now, a power law in statistics is a functional relationship between two quantities where a relative change in one quantity results in a proportional relative change in the other quantity, independent of the initial size of those quantities. One quantity varies as a power of another. For instance, considering this area of a square in terms of the length of its side, if the length is double, the area is multiplied by a factor of 4. So what that tells us is that very often differences in sizes of epidemics in different areas appears to be best described as a rough power law equation. The other interesting part is that it is at this tail end of the power law distribution that we see sometimes an enormous amount of variability that can introduce substantial differences in how the epidemic behaves in different areas in different periods of time. To illustrate this, let us look at a sensitive dependence on initial conditions, which is the Lorentz equation. So this Lorentz equation, we have three different conditions which have very minor variations, where 1.001 is compared to 1.0001 versus 1.00001. And as you can see, within 12 seconds, the curves start diverging. So a very small difference in the initial conditions can actually lead to substantial differences in the eventual outcomes. This is called a sensitive dependence on initial conditions and is a feature of many epidemics, particularly in the tail end of the power law distribution. Now, epidemics also demonstrate what we call a complex adaptive behavior, where within the entire system there are multiple different agents that are interacting with each other, and there is external influences that come in to bear on the system. There are simple self-organized local relationships, there are feedback loops, and sometimes there are what we call strange attractors that pull the entire system one way or the other. The Lorentz equation impacting on the tail end of a power law distribution would be an example of a strange attractor here. So this is very important to understand, that predicting exactly how an epidemic will behave or epidemic projections can sometimes be rather complex and not easy to understand, describe, or predict mathematically. Now the key question is, COVID-19, was it man-made? And in particular, all of these variants, are they man-made? So this question has been raised. Is COVID-19 a consequence of something that happened inside the lab of virology at Wuhan in China? And why did it happen? Was it deliberate? Was it inadvertent? Was it a consequence of gain of function research? And how did all of this happen? Was there some incompetence on the staff? Was this unethical behavior? And then what happened? It led to a global pandemic, enormous socioeconomic impact, but just as importantly, a loss of trust in scientific research. So how have these questions come up? So it is important to understand what is gain of function research. And this came to the fore particularly because of a rather heated debate between Senator Rand Paul and Dr. Anthony Fauci. By the way, Senator Rand Paul is also a doctor. And the concern that was raised arose from a 2017 paper by the scientists at the Wuhan Institute, which had been supported through NIAID, which is a part of NIH, through a New York-based organization called EcoHealth Alliance. And it had been done at a time when funding for gain of function science research was supposed to be barred. The authors genetically crafted spike proteins, the viral keys that grant access to mammalian cells, from 8 different naturally occurring coronaviruses into another coronavirus from the wild called WIV1, and found that these new creations in lab dishes could infect monkey kidney cells as well as human cells through the same gateway, the widely expressed ACE2 receptor that is used by COVID-19. Now, unfortunately, the research is not properly understood by many people, and a lot of what I would consider unfair blame was placed on Dr. Peter Daszak, who is the president of EcoHealth Alliance, which is a US-based organization that conducts research and outreach programs on global health. They have been instrumental in doing a lot of surveillance, coming up with early warning systems to warn us about zoonotic infections that can spread to humans, particularly identifying the bat origin of SARS the drivers of Nipah virus emergence, and publishing the first global emerging disease hotspots map, etc. Now, this is incredibly important research, and really Dr. Peter Daszak and EcoHealth Alliance have been instrumental in alerting the entire world to the dangers of these kinds of infections spilling over from natural enzootic reservoirs into human populations, causing major disasters. Unfortunately, because of all of the loss of trust and the acrimonious debate about gain-of-function research, Dr. Daszak and his organization was unfairly targeted, blamed for all of this gain-of-function research, and the messenger was shot. Now, coronaviruses is something that we have known about for a very long time. In fact, the first discovered coronaviruses were infectious bronchitis viruses that cause respiratory disease in chickens, and this was discovered in the 1930s. So we've known about a lot of coronaviruses for a very long period of time, including human coronaviruses 229E and OC43, which cause 1/3 of the common colds in humans. SARS-CoV was discovered in 2002, NL63 in 2004, HKU1 in 2005, the Middle East Respiratory Syndrome virus in 2012, and in December of 2019, we saw COVID-19 appear. So we've known about coronaviruses for a very long time. We've known about the basic virology of these viruses. We've known about the basic pathophysiology of these viruses because of the studies on animals as well as humans with previous COVID infections or previous coronavirus infections. Now, one of the key things to understand is, based on the work that Manfred Eigen, a Nobel laureate, did with mathematical modeling in the late 80s and early 90s, that viruses essentially are a quasi-species, which means that an entire viral population demonstrates a tremendous amount of heterogeneity, and very soon we will see the emergence of multiple different genetic variants. So this is a predictable natural occurrence of many pathogens, but in particular virus species that replicate at enormously high numbers. So this is part of the reason why it is entirely predictable that COVID would develop all of these variants, alpha, beta, gamma, delta, and so on and so forth. Now, when we look at the genetic heterogeneity and the accumulation of various mutations in Delta and Omicron, we find is that there are numerous mutations that occur as compared to previous variants. But in particular in Omicron, there are a larger number of mutations, particularly in the spike protein area, which is the receptor binding domain, as compared to Delta. There are many more mutations, but not only in the spike region, but also other parts of the virus. This is a study that Venki Soundarajan and his associates have put in a preprint version. Very interesting data. And in particular, if we look at the difference between Delta and Omicron, we see that there are almost 26 odd mutations in the receptor binding domain. And the significance of this is that it may be the reason why Omicron is able to evade immune responses produced by previous infections of coronaviruses, as well as vaccinations that depend primarily on response to the receptor binding domain, such as the mRNA vaccines that are most popularly used. Also, it is possible that antibody cocktails that we use for the Delta variant so effectively may not necessarily be as efficacious with Omicron, because the receptor binding domain has so much more genetic heterogeneity as compared to the Delta variant. Now, if we look at the phylogenetic trees, what is apparent is the Omicron strain is so very different from all of the other variants, alpha, beta, gamma, delta, and so on and so forth. It's almost as though this is an entirely new species of virus, but not quite. Now, why does this happen? Why do we have Omicron that is phylogenetically so very different from the other variants? And this is important to look at the explanation for this, because that lies in understanding the basic virology. Now, when we look at coronaviruses, once a virus is seen, there are many different parts of the virus, and what I would like to draw your attention to is the spike protein, which is the receptor binding domain, but please also pay attention to the envelope glycoprotein E, because this part of the virus is also very important in the pathogenesis and the pathophysiology of these viral infections. Now, once the virus has attached to an ACE2 receptor through the receptor binding domain, which is the spike protein, it uncoats and then it is replicated by the RNA-dependent RNA polymerase. But in addition to a complete replication, it also replicates into smaller mRNA fragments that translate into the different kinds of proteins that are made by this virus, important in the virulence and the pathogenesis. Now, the importance of understanding this is that these smaller mRNA fragments have the ability to recombine, reassort into many different variants. So despite the fact that coronaviruses have a proofreading exonuclease, the incredible amount of replication and as well as the reassortment and recombination of multiple fragments of the virus result in a tremendous amount of genetic heterogeneity, far greater than the ability of the exonuclease to proofread the progeny viruses. Not only that, the ability to recombine and reassort means that they can also recombine and reassort with different viruses, not necessarily the parent virus of COVID-19, but other similar viruses, and then come up with totally new kinds of viruses. And let's see how this has happened in the case of Omicron. So once again, this is a study from Soundararajan and his associates, and what they postulate is that inside an infected cell, we had two different viruses. The human coronavirus 229E, which is responsible for causing common colds, and then a Omicron predecessor, which is probably a Delta variant of the SARS-CoV-19 virus. And because of what we discussed previously in terms of reassortment and recombination, fragments of these two viruses have recombined to produce the Omicron variant. Now, this probably occurred in an immunocompromised individual who had ongoing persistent replication for prolonged periods of times, which would vastly increase the probability of such a kind of dual infection and recombination reassortment, leading to a completely different kind of virus. But this was again something that was predictable because of the inherent nature of coronaviruses that replicate into such high numbers with such great genetic heterogeneity, and the inherent ability for recombination, reassortment of fragments of viruses that lead to even more genetic heterogeneity. So this was an entirely predictable outcome, and that it has happened should not surprise anybody. Now, let us look at what is happening in terms of the cases and deaths. And as you can see, that there seems to be in this area a little bit of a divergence in the number of cases and deaths. There are far more cases and fewer deaths as compared to the prior periods of time. And the reasons for this may be manifold, but part of the reason may simply be that Omicron does not cause as severe an illness in infected individuals. But in addition to that, it is also possible that by the time Omicron hit, the population also had some amount of resistance or immunity to various viruses. Now, despite the fact that there is an enormous difference in the receptor binding domain, and perhaps the receptor binding domain antibodies may not work as well in the case of Omicron, that is not necessarily the full answer, because as we can see, there have been a large number of people who have been vaccinated, more in the United States than in India. And also in the United States, there are a lot of people who are receiving booster doses. And what this does is it actually increases the amount of antibody produced after a booster dose in people. And this is particularly true of the mRNA vaccines. Now, in addition to higher antibody titers after a booster dose, one must remember that antibodies are not the complete answer in terms of protective efficacy against COVID-19, or for that matter, any virus. And here we can see that in anybody you have innate immunity, and then you have adaptive immunity, which is both antibody mediated as well as cell mediated. And what is interesting is that sometimes innate immunity can be trained by exposure to heterologous pathogen receptor entities that are different kinds of pathogens. For example, it is well known for many, many years that prior immunization with BCG actually for a short period of time gives a protective efficacy against many different viruses. This is a well-known phenomenon that has been observed many, many years ago. And also, experimentally, we can see that different kinds of vaccines, such as MVI-130 and MV-140, provides heterologous protection against a variety of different pathogens. So what this means, the significance of this is that even innate immunity can be primed by other stimuli to produce a robust early interferon response that might help to attenuate the infection by a heterologous pathogen. And also that it's not simply an antibody response that is important. Cell-mediated immunity is also important in the adaptive response, and this cell-mediated immunity is not necessarily directed only against the receptor binding domain or the spike protein, but may be directed against many other parts of the virus. And that is maybe part of the explanation for why we see that even in the case of Omicron, in people who have been vaccinated against the other variants, Delta and so on and so forth, that there is a protective efficacy in terms of reduced morbidity and mortality. Now, the other part of the virus is, which is very important, is Viruporin E3, because Viruporin E3 is probably the principal part of the virus responsible for inciting an inflammasome known as NLRP3. And NLRP3 in particular is implicated as one of the major factors in the inflammatory response to COVID-19, as this cartoon shows. NLRP3 increases neutrophil infiltration, it increases fibrosis and severe lung damage. It will also upregulate high mobility group box 1, which is a danger associated molecular pattern implicated in severe macrophage activation, and all of these eventually lead to a cytokine storm. So NLRP3 is clearly a major attribute of the cascade of responses that result in severe inflammatory responses. Now, Viruporin E3 in particular is responsible for upregulating the NLRP3 responses and the inflammatory cascade. So in those individuals who have been vaccinated and have the ability to block Viruporin E3 mediated responses, they would naturally be protected. So again, I go back to reminding ourselves that it is not merely the spike protein that is important, other parts of the viruses are also important. And this may be part of the reason where antibody preparations like sotrovimab, which has neutralizing activity against the entire virus, may continue to have efficacy even against strains like Omicron, which have substantially greater differences in the receptor binding domain, which might reduce the efficacy of antibody cocktails directed against the receptor binding domain. Now, why are coronaviruses not lethal for bats? As we can see in this slide, there are many different coronaviruses that are present in bats, and they infect a variety of different animals, including humans and birds, but they can be lethal in all of these other species, and yet the bats go around merrily living with all of these coronaviruses. In fact, a variety of other viruses as well, such as Nipah virus or rabies viruses, and they seem to do quite well with it. And here the reason appears to be that in bats, there is a dampened response to NLRP3, which we previously discussed as very important in the pathogenesis of the severe inflammatory response to coronaviruses in human beings. So this may be part of the explanation for why bats can remain enzootic for all of these different kinds of viruses, not have any problems with it, and yet these viruses, when transmitted across the species barrier to other animals, can be deadly. Now, in conclusion, what do we need to address the Omicron, or in fact the entire COVID-19 pandemic? We need to continue our emphasis on non-pharmacologic interventions such as physical distancing, but just as importantly, ventilation and vaccine, antiviral agents, and credible clinical care. The only way we will make a significant change in the epidemic dynamics is the ability to vaccinate a great deal of people. So in this complex adaptive system, which is the epidemic, the strange attractor that we need that will move the entire complex adaptive system towards better control is something like vaccine, effective antiviral agents, and then credible clinical care. We also need to remind ourselves that in India, we need to consider the socioeconomic conditions. 65% of the population is rural with negligible access to medical care, not enough public hospitals, and because of all of the reasons that we discussed previously, in particularly high transmission rates, cohorting a large number of infected people in small areas is not a good idea, because that would vastly increase the probability of greater transmission and greater emergence of even more variants that may be different in how they are transmitted and also different in morbidity and mortality. They may be milder, but they could just as well be much more serious illness. So we need to have a plan to address the economic impact of illness on the patient and the family, the socioeconomic impact on various restrictive measures in movement and economic activity. We need to consider the long-term complications of COVID, lung damage, emerging infections, particularly tuberculosis and fungal infections. Inappropriate treatment regimens for COVID should be avoided, particularly overzealous use of high-dose steroids, inappropriate use of immunomodulators, ineffective agents such as remdesivir, ivermectin, or convalescent plasma. And we also need to have the ability to help people who have long-term disability caused by COVID-19 and a consequent loss of ability to earn a livelihood. But most importantly, I think we need to remember that One Health and the concepts of One Health are very, very important if we are to ever prevent something like COVID-19 happening again. It may be yet another pathogen, a different pathogen tomorrow, which will again cause the same kind of severe socioeconomic impact due to a global pandemic. So COVID-19 is a warning from Mother Nature that we ignore at our own peril. So as Gandhi said, be the change that you want to see in the world. Power is an illusion, authority is a delusion, responsibility is reality. All of us need to understand our responsibility and think about what we can do individually as well as collectively to address the COVID-19 pandemic, and what is our responsibility to ensure that we build into our healthcare systems and our society the kind of resilience to avoid deleterious impacts of other potential pandemics in the future. Thank you very much.